Or you stay home. I am. I like. Nice.
Really? The why there's some fish game.
No.
Like he's more like.
I think we have.
Yeah.
See what I think.
All right, shall we get started? Okay, cool. All right. So this is my great pleasure to have Ratan Murty here, speaking to us about towards the computational intelligence. So Ratan is very new to the Georgia Tech neuroscience community. So he's an assistant professor in the School of Psychology, started this January. Before coming to Georgia Tech, he did his Ph.D. at Indian Institute of Science in Bangalore, and then followed by a postdoctoral training with Nancy Kanwisher and Jim DiCarlo at MIT. He has won like various awards, including K99 and NIH Young System Scientist Award. And yeah, thank you for being here.
Can everybody hear me okay? Great. It's really nice to be here. Thank you, Hannah, for inviting me. Now, this talk is meant to be really broad. And what I want to actually convey today is that we are stepping into this new way of doing science, especially in human systems neuroscience, human cognitive neuroscience. But before that, I really want to start by telling you a little bit about vision, right? And I think the thing to note about human vision is that it is just surprising how well it works, right? For instance, all you have to do is close your eyes and open your eyes and just see this scene, and you can very easily tell where you are, how you might navigate in this space, what are all the things that you might want to avoid, okay? And then you also use vision for other kinds of stuff, right? Like, so for example, to identify people, to see what mood they're in, right? You can look around and I can very quickly tell how many of you are actually paying attention versus eating your food and so on. And this happens pretty instantaneously, right? We also use it for other kinds of things like social interactions. So just looking at somebody, two people speaking, you can tell whether there's a positive interaction or not. And we do this all the time. So let's look at the brain. And if you look at the functional organization of the human brain, you see a lot of order, right? And it doesn't matter which part of the brain you work on. As soon as you look at how the human brain is organized, you see a lot of systematic regions, parts interacting with each other. And if you talk about the visual cortex more specifically, you will find that there are all these regions that seem to do one thing. Okay, so very broadly, if you've taken a human cognitive neuroscience course, you know this. If you look at just the bottom half of your brain, so there are parts that seem to respond when you look at images of faces, images of scenes, bodies, recently we're also sure it's probably also images of food, all those things that you're eating, right? Um, and these regions are all present in each one of you, right? They seem to land in approximately the same location. And that raises a series of questions that my lab is very, very interested in. And let me first lay out the questions that we like to look into. So the first question that you might have is, if you have the systematic organization, what is going on in the brain? What are the kinds of features that those parts of the brain like? And how are they encoded in these parts of the brain? So I'm talking about high-level vision for now. So you might be wondering, hey, what's going on? Like, what is it about that face? Or what is it about the scene that is really driving these responses? And why does that happen, right? The second kind of question that you might have is, if you're seeing this structure emerge, how does it get constructed over development? What are the factors that are necessary? How early is it that you have these things, right? And are there canonical computations that you might be able to use and co-opt for other computational systems? Think about AI or any other field that might be of interest to you. Now, if you are working in humans, which I am, this is another thing that you might really care about, which is what would you do if typical neural mechanisms go awry, right? So this is really, so my background is actually in studying macaques, and when I moved to studying humans, this was the thing that was really motivating my research, is that if you want to understand the mechanisms of neural disabilities, then you've got to at some point contend with human brains. And this is a big thrust for what the lab intends to do in the near future. And the fourth thing is really, we're talking about the construction of the human brain, but it's hard to talk about this in the absence of any kind of behavior, right? And I started with behavior, but I'm not going to talk about a lot of behavior in today's talk. Just to say that there are a number of things that we've been working on that span this entire space. I can give a talk on each of these things for an hour, but for today's talk, I'm going to keep it super broad and tell you that the core way that my lab does science is by the development of really accurate models, right? We're going to be using computational models to understand how the brain works and answer all of these questions. So that's in the very high-level gist of what the lab is doing. Okay, so the first question that you might have is, okay, why are we talking about models, right? This has nothing to do with biology, and I want to motivate that for a minute. Okay, so this is the bottom view of a brain. And if you haven't recognized this yet, this is like the bottom of view of my brain, right? So now, if you look at this part here in yellow, that's what's called the fusiform face area. So it's a part of the brain that's been extremely well studied. And if you look at a typical set of responses in this part of the brain, this is what you find. Okay, in case you haven't seen this before, on the x-axis, by the way, is images. On the y-axis is how strong this part of the brain responds to these set of stimuli. Okay, everybody on board so far? The color is indicating which kind of stimulus was presented to a human being, right? So every time you see something like blue, it's an image that contains a face, right? Okay, so it's data like this that have been used to, you can take just the first principal component of this data, high response to face, less response to other things. We are going to call it a face region, right? So that's the kind of stuff that's happened for a while. And people would conclude that, the responses of this part of the brain are faces, right? Now, the central motivation of my work is that this inference is actually highly imperfect, all right? There are at least 3 issues with this central idea. Okay, let me spell out what those issues are going to be. The first issue is that just concluding that a part of the brain is selective to faces massively underspecifies the kind of response, the richness of the response that you get in this part of the brain, right? So what do I mean by that? It does not explain, any of these stable differences. And by the way, these differences are very, very stable, right? You see them in almost every participant. So if you truly understand what this part of the brain does, you should be able to explain what's going on here. But this account, that a part of the brain is face selective, does not take that into consideration at all. All right. Here's the second issue, that you always need a human in the loop, right? If I tell you that a part of the brain is face selective, like it seems as if you've understood something, but you can't make do with that knowledge at all. So for example, if I tell you a part of the brain is face selective, and some of you maybe have heard this idea for the very first time, and I say, okay, what's now the response of this thing in my hand? You have no idea. It doesn't tell you very much about what other things might be, and doesn't make predictions for any stimulus. So that's the second point, that issue that this particular inference has. And this is, by the way, a general theme in a bunch of work that's happened in cognitive neuroscience. Okay. The third part is a little bit more controversial. All right. And the third part is the follows. How do you really know that the idea of category selectivity is even true? If you think about this idea is an inherently reckless hypothesis, right? Which can very, very easily be refuted. If you say, for example, you record for an image of a pineapple someday, and you find that this pineapple has a higher response to, say, the image of my face, this entire gig is up, right? So this area is no longer category selective. Now think of the number of experiments that might have been performed on the FFA, right? If you think, okay, maybe 1,000 studies, they would have evaluated what, 1,500, 2,000 kinds of stimuli, and that's it, right? All these inferences are an inductive leap from the set of studies that you've performed, that you're generalizing to the entire class of stimuli in the world. And that's a large set, okay? Now, the reason why I started to introduce these ideas was that there's a very easy way to solve all of these problems, which is that if you have computational models that make accurate predictions, you're already kind of solving for a bunch of these things. So here's a model desiderata, right? Like here's what we want. We want to generate models that are highly predictive, because prediction matters a lot, especially in this field, right? Then you also want models that are also image computable. By that, what I mean is that you should be able to take any stimulus that you have and be able to run inference on the model. So that's the general idea. And now here's the third thing, which is actually something that people care about a little bit less, but I think is super important, that you want models that can also interface with everything that we know about from cognitive science and psychology, right? So I'm going to come to this part in the second half of my talk. And let me first start by showing you how good these models can be. Okay, so this is a study that I did back in my postdoc. This idea is kind of very, very simple, which is that if you look at the kinds of representations that you get from many kinds of neural networks, these neural networks actually are very rich. The representations are quite rich. So you can actually create what's called an encoding model. The idea is that you can show humans a bunch of stimuli. You can take the same stimuli and run them through a model, extract some activations, and map that to the brain. So that's the general idea. Some of you may have heard of it. If you have not, it doesn't matter. The goal is to show you how good these models can be, right? So you can evaluate the goodness of the model by estimating some kind of test or correlation between the predictive response and the actual measured response. So I'm going to show you that next. On the x-axis here is the predicted response. On the y-axis is the observed response. And every single dot is going to be a single stimulus. And I kind of gave it up very early on, but what we find is that the correlation between the observed predicted response and the observed response is quite high. In general, what you find is that even for stimuli that are within faces, which are shown here in blue, so this is the response of the preferred category, in this case, faces, you can predict significantly better than anything that you had previously, right? So that is also true for whatever the other region, also true for other parts of the brain. So I'm going to show you now responses for two other parts of the brain. This is a region that's more selective for bodies. This is a region that is more selective for scenes. it doesn't matter where you look at, you're able to predict responses with very high accuracy. Okay, so the punchline here is that wherever you look at, based on some fMRI data or single neuron data from humans, you do reasonably well at predicting responses to stimuli that the model's never seen before. So critically, the model has never encountered these images when it was being trained. Okay, everybody on board so far?
So the predicted response here, this is like an average over the voxels in that region, or is it like the pattern similarity?
We've done both. So in this particular study, what I'm showing you here is the voxel average response. And the reason we do this here, by the way, is because we have no hypotheses about a voxel in the FFA. But we have a hypothesis about the entire part of the FFA. But if you look at what happens, like in this particular study, what we did, we looked at individual voxels, we looked at the representational geometry. You can take any measure, and you find that you can predict it really well with neural network-based encoding models. Okay, so let's go back. Now, I just showed you that the models are pretty good. What I've not shown you here today is that they generalize really well across subjects. So you don't even need the same subjects on which you train these data. So that's the general idea. Now, when we originally published this paper, the question was, okay, now we have a good model. We think we have a good model of the FFA, but better compared to what, right? So we are saying that I have something good, but the problem was that, the previous model of the FFA lived in professors' heads, right? So because that's the understanding about the FFA. We got professors to make some of these predictions, and they were terrible at it, right? So it turns out that you can predict brain data better than any other person out there and any other system that we had previously. So that's the general idea. And if you go back to the kinds of things that I raised in the first part of the talk, just having predictive models solves at least two of these issues that I had raised, right? So you no longer need to worry about like individual images because the models can predict really well. And you no longer need a human in the loop anymore. So you can actually run any stimulus and get a prediction. Okay, so let's go to the third part. Is category selectivity even true? Right? So there are other ways of doing this. We showed several ways here. But let me give you a kind of method that is really scalable to other problems that you might have, which is to take a model. In this case, that was the model that I first showed you. So that's the model coupled to the brain. And you can actually append what's called now a generative model, right? So this is when we were doing this experiment, the best generative models out there were what's called GANs, which is generative adversarial networks. Now people have shown this with, you don't even need this kind of image prior. You don't even need, you can use a diffusion model because that's the state of the art today. And you can run this whole inference pipeline through a generative model like a GAN. And you can ask the model, okay, tell me what are the stimuli that are really supposed to drive responses in this part of the brain, right? So why is that interesting? The reason is that the generative model can actually come up with almost all kinds of naturalistic stimuli in the world. And you can push this model completely, like entirely forward and see what the model is really like. Okay, so you may have heard the talk of like Carlos Ponce a few weeks back. And when he was showing this kind of approach, like they were using a method that's called a genetic algorithm. Note that this method doesn't have a genetic algorithm at all. So it's not that you're doing a closed loop with the brain and getting a better response. This is a one-shot prediction of using a model to tell which kinds of stimulus properties the models really like. All right, so that's the general idea. And what happens when you do this is these are images that GAN produces that are predicted to drive responses in this part of the brain. That's for the FFA. Those are kind of images for the EBA. And these are images for the scene regions or the parahippocampal place area. And I'll just fill this up with other parts. So you can just very quickly appreciate that these are unambiguously faces, bodies, and scenes. Okay. So far, I've not told you anything different from what we know about these regions, right? But to show that this is a test, you can think of this as a test case for what is possible now when you go outside these parts of the brain that have been relatively well characterized. All right, so I've shown you that you can predict responses. And once you have a very good predictive model, you can go back to the explanation space. right? So you can take a predictive model and you can start reverse engineering what are the kinds of selectivities that drive the responses. But you can do a little bit better. What you can also do is you can go inside the model and ask, okay, like what are the specific stimulus properties that are really driving the responses in this part of the brain, right? This is an experiment that was possible in macaques previously because you need a large number of stimuli to really figure out the kinds of stimulus properties that drive responses using methods like reverse correlation. That was never possible in humans because you really can't subject humans to many hundreds of stimuli. But now with a predictive model, you can go in and identify the specific stimulus patterns that drive responses in these parts of the brain. Okay? All right. Now I'm going to start putting all of the core results from this talk on a slide that looks like this. So far, what I've shown you are, if you have good predictive models, that gives you some kinds of superpowers that you did not have before, right? That's how you should think of these results. The first thing that I showed you is that you can use a predictive model to reverse engineer what that part of the brain might be doing, right? The second thing that I showed you is that you could then use the model to also find the features that could drive responses in this part of the brain, right? I hope to cover 2 more sections in this particular talk. In the next part, I want to ask another kind of question, which is, can we use models to bring some kind of computational precision to the classic theories of vision, right? Because vision is a very old field. Like there are several theories about what different parts of the brain do. And the question is, like, can these models even inform those theories or even better improve upon them, right? So that's the general idea. Okay. Now, if you've just seen these results so far, all of you should be really skeptical, and I hope you are, right? There are several reasons why you should be skeptical, and I'm going to lay out at least 2 reasons why. The first is that the inference that I'm showing you, we really just showed you results for like 1 data set, right? And that's not very much. That's the stuff that we collected in the lab. The other issue is that we don't know where these models are going to break, right? So we don't have any kinds of bounds, right? Like all models have bounds. I mean, Newtonian mechanics have bounds, right? So we don't know what the bounds of these current models are. So the question is like, how do we make progress? Now, the third thing that I think should make you really skeptical is like, let me take a minute to like articulate like what I am really telling you so far. So the claim is the follows, right? Like, so there's a part of the brain that each one of you has. I can find it in 10 minutes, right? That's all I need to find that part of the brain. You can see any image in the world that you like, right? But the claim that I have is that you can then take that particular part of the brain and I can simulate it on my laptop, right? So that's kind of a ridiculous claim, right? Like, how many things that can we even test? Like, how do we know that these models are really good, right? We don't, right? So we got to do a little bit better. All right. So the other question that you might have is, are we done? If we have predictive models, there are other kinds of things that have been raised as issues related to, just in general, the use of neural network models in vision. These models that I showed you do not have any recurrence, for example. They do not have any kind of biophysical principles, right? They are just feed-forward systems doing, very few canonical computations. That's all that these models really have. All right, I'm going to skip a few things here and tell you our strategy now. Okay, so in the broad sense, what we ask is, okay, like how many of the previous studies can these models really recapitulate? So that's the general kind of idea. And this is work that's being led by 2 really nice, good students from the lab, Alex Abate, who is now at Harvard Medical School, and Roland, who is a postdoc. And let me just describe the procedure from scratch. Okay, so the idea is that there are several studies that you can take from the field, right? That's easy. You go on PubMed, you can just type the fusiform face area or any other part of the brain that you like, doesn't matter. Now, the things that you can do now is you can ask whether you can recapitulate those exact same experiments on models of the brain, right? Here's the process. You can take, I'm going to call them virtual FFAs. So this is like a model of these parts of the brain. you can take that experiment and run it exactly as the original study, but this time on models of the brain, right? And then what you can do is you can chronicle like how many times you succeed and how many times you fail. So that's the general strategy. That's the heuristic, right? Now, turns out that when you think about this, you run into some kinds of problems, right? I'm going to call fun of the first version of this qualitative comparisons. The reason I'm going to call this qualitative is there are two kinds of Turing tests really that we are running. Okay, the first Turing test is the follows. Imagine that you take a study in the field that you really like, that's performed on this part of the brain, right? You can run that same experiment on the model of, the FFA, for instance. And at the end of that, you should ask yourself, okay, would I write the same paper as the original study? Okay, so that's the first kind of Turing test, which is like, will I write the same paper? The answer could be yes or no. I'm going to give you some examples so that it gets a little bit more clear. The problem here is that people have not really shared their data, so we can't really make quantitative matches. So that's the general issue that I'm trying to highlight here. The second problem, the second way of comparing data can be just quantitative, right? Like, so this is stuff that we have out there, open data sets. We are in a new world. Things work really well now. People share their data. Awesome. So we can do that as well. So these are the two kinds of tests that I'm going to briefly show you today. All right, so let's start with some qualitative comparisons. Okay, I'm going to take one part of the brain and show you many different kinds of studies that have been performed on this part of the brain, right? All right, so if you know something about face processing, you will recognize some of these studies and you'll realize that each study is going beyond just telling that the face region responds more to faces. It's trying to encapsulate an idea, a cognitive variable that this part of the brain might be responsive to. Look, so for example, you can take something like the face inversion effect, for instance. And in this idea, what people have seen is that if you take inverted versions of faces, then they seem to have a significantly lower response in regions like the FFA, which presumably is the basis for all kinds of illusions that you have, the most prominent of which is the Thatcher illusion, right? So if you take an inverted face, you don't perceive it as well as you perceive upright faces. In this world, we see upright faces way more than we see inverted faces. That's the general idea if you think about it from even like an information theoretic kind of perspective. All right, so now the question is, okay, if we run these experiments on the FFA, why do we even expect failures, right? And instead of me just trying to articulate why we should expect failures, I want to show you how creative some of these experiments really have been, okay? So in order to demonstrate that, I thought I'll take a study that was done here in Atlanta. So this was done from some of our colleagues over at Emory. The title of this paper, which unfortunately you cannot see, is that the faces is more than the eyes and the nose. Okay, so how do you test this hypothesis in psychology and neuroscience? Here's what you do. Okay, you extract the internal features. In this case, you extract things like the eyes and the nose and the mouth, and you present them to humans. Here's another condition where you remove the eyes and the nose and the mouth, and you just show the outline, right? So you're removing the internal features, removing the external features, and then you present it against two other conditions. Okay. Now, the reason why this is interesting is that these are the kinds of images that the models have never seen. They've not seen this while they've been trained. They have not seen this when the models being fitted to these data. So this is a strong test, I would argue. All right, so what do they find? So this is figure 2 from their study. And in general, what you find is that there's a higher response to the internal features relative to the response to the external features. And then you have relatively low response for these other categories, as you would expect from a region that is selective to faces, right? So that's the general idea. So that's what you find. All right, so what happens if you run the same experiment on the model of the FFA? You get pretty much the same pattern. You would write that same result, you write the same paper, except that, I was doing a back on the envelope calculation with Danny. They spent about $18,000 running that experiment. We can run this in a minute. All right. Okay. So some of you may not care about just voxel average responses, and or you asked that question early on. I mean, there's actually way more to what these regions do than just averaging responses, and that's 100% true. So I want to give you another experiment just to illustrate the point that you can run the same experiments even in high dimensional spaces, multivariate patterns of response. Okay, this is a paper from the NIH, from Chris Baker's lab, and they're studying the response to images that have face pareidolia. So if you don't know what face pareidolia is, it's this perceptual property of seeing faces in everyday things, right? I hope all of you is seeing a face in this cup of coffee. If not, reach out. And then what happens, so in this study, what they do is they compare the response to objects, which is the same category as, and the same thing as the original stimulus, and then to other objects, right? So that's the general kind of thing that they present. Okay, so the way they show these data are by running what's called a decoding analysis. So some of you are from machine learning and other, so you know what this is. But on the y-axis here is going to be the classification accuracy. Okay, and they're doing pairwise classification. So if everything was just chance, your accuracy will be close to 50%, right? And if you've not seen these kinds of plots before, the way you should think of this as follows. So if you have two patterns that are very similar to each other, you cannot read out between them. So the lower it is, then it means that the two patterns are very similar to each other, right? And the higher this number is, it means that the patterns of responses were quite dissimilar. That's the general idea. All right, what do they find? I'm going to run this animation through so that you understand the plot a little bit better. What they find is that illusory faces and objects are actually more similar to each other, which is why I put them close by, than human faces. Illusory faces is somewhere between human faces and objects, but closer to objects. So that's the general pattern that you find. Now, if you look at the same result on the multivariate pattern plot, this is what you will see. This is the dissimilarity between illusory faces and objects, which is low, which means that they are more similar to each other. That's the pattern, okay? I hope all of you got this plot. All right. Okay, what happens in the model of the FFA? Turns out you get really much the same pattern of activation, right? One thing you should note is that the y-axis is significantly different, right? Like you see this y-axis go from 0 to 100%. And that's because the y-axis here depends on the quality of data that you have, right? So if your data were really good, as they were when we were building these models, then the decoding accuracy is going to be quite high. But the question is, would you write the same paper as the original authors? The answer is yes. Okay, I'm not going to bore you with all of these experiments. Believe me when I say all of these studies, every single one of them can be replicated on the model today, right? Okay, so based on that, you can conclude, okay, like we have very good models of the FFA compared to anything that we had previously. Okay, so recently what happened is, maybe some of you may have heard of this. There's a new data set that's called the natural scenes data set. The general idea is the follows. You can think of them as, this is a new scanner, different subjects, different images. We know nothing about these stimuli. The model's never been trained on this. All of our predictions are zero shot, right? We don't know, and that's why this is from Minnesota. And so there is this data set that just came out. So you might ask, okay, like how good are these models at just predicting these data without ever having encountered any of these stimuli? So that's the general idea, right? Okay. Here's how you evaluate the models. So now on the y-axis is the model prediction accuracy. What I'm showing you here is the noise ceiling of the data. But noise ceiling is not computed over fMRI data. So the way you should think of this is how similar are two humans to each other, right? So if you and I are quite similar to each other, that's going to be a measure of similarity across brains. So just a reminder again, we are thinking of this as Turing tests, right? So you can ask how similar two humans are to each other, and then you can ask how good a model is relative to another human being. So is it just like another human brain, or is it more like an alien brain, right? So that's the idea. Cool. All right. How good do these models get at predicting these data without having seen any of these stimuli? That's how good the models are, okay? Turns out it is very close to the distribution of the stimuli that were run in the natural scenes dataset. So you can predict almost all the variants of the natural scenes dataset for these parts of the brain without having trained the model on any of these stimuli. Entirely zero shot. Okay, so here's the other thing that we've been doing is we are trying to come up with a completely executable framework. So this is something that all of you should be able to try, right? You can run this on your machine today, right? So here's a general idea. We are providing people ways of running your own experiment without even, you don't have to share your data with us. You can run inference on these models without even having to discuss it with anybody else. You can request for all kinds of, data. If you like univariate analysis, you can take univariate data. If you like multivariate analysis, you can do that. It doesn't matter if you like MATLAB or Python. Everything is GUI based. You can run this today. This is available right now, right? So in general, we call this framework the idea of like having a virtual visual cortex. The goal is to create something that's entirely executable to simulate, your own prior experiments. If you've not shared your stimuli online, you should be able to run this yourself. Or even, so you can simulate your future studies today without having to run a single study, single subject on the fMRI scanner. In my mind, we have three big goals with this effort, right? We want to make sure that we are able to organize everything that we have, all the frameworks, all the models, all the data in one place, right? The other, of course, is a scientific hypothesis testing idea, which is that we want to make sure that people in schools of psychology who do not typically work with computational models have a way to test their hypotheses directly without having to interact with any code whatsoever. Make it as easy as possible for them to do their science. So that's the second idea. And if you have the third idea is, of course, for access and teaching. My students in my class, some of whom who are here, already use this for different kinds of assignments. They can run their own curiosity experiments on models of the brain without, and that's actually been a really fun thing to do. So this entire framework is going to be very quickly available. The goal of this entire procedure is to actually really lay bare what we think are going to be the strengths and limitations of current models, right? The issue right now is that, of course, we think that the models are really good, but you can imagine this is what happened when OpenAI was releasing the first version of ChatGPT, or when Google was releasing their first version of Gemini, right? Internal teams would think that they're really good, but as soon as we make it available to the public, that's when the rubber hits the road. That's when I hope that a bunch of cognitive neuroscientists come out and say that, here's a specific experiment that you should not be able to predict, or, this is a version that where your models are broken. The goal here is really to figure out where models fail. And we're trying to crowdsource that effort of model failure, not success. Okay, I think I already made that point. So the other thing that we really want to do is we want to make sure that this cycle, which is what I think of the experimental cycle of science, which is you start from an experiment, you develop a model. People call this model a hypothesis many times, but really it's a model, right? And so the way it works is you want to hasten this cycle between experiments and models, right? And that's what having an open platform like the virtual visual cortex might enable us to do. We supply the GPUs. If you're interested in testing, you can reach out to us. Okay, right. So now I want to go back to the top level questions that I had. Now, I think of what was happening in cognitive science recently is that we were collecting a lot of facts about what the human visual system responds to. And we are now trying to come up with a common framework that can explain a large number of these properties entirely zero shot, right? And turns out these models are really good at recapitulating a bunch of experimental results from prior studies in the field already. And then I wanted to briefly introduce you to this idea of the virtual visual cortex, which is this idea of having an executable platform for you to simulate your experiments today. Okay, how much time do I have? 10 minutes?
20 minutes till we need to be finished.
All right, okay. Maybe I'll do this next part. I think it's cool. All right, so the next part, what I want to show you is what else can we learn? Can we use these models for things like, discovery, scientific discovery? So that's the third part of my talk. And for that, version of my talk, what I want to focus on is like, how we really think of as experiments in neuroscience. And this is again something that Audrey really referred to early on, is there's a kind of hypothesis that many people have when they talk about fMRI data, which is that all of these boxes, which I'm trying to show here in black, are kind of doing the same thing, right? This is, I'm taking one part of the brain, this is called the parahippocampal place area, and you would look at this brain area and say that, hey, you know what? Let's assume that they're all the same thing and then average over those responses, right? It turns out that it's not that easy. So if you look at what goes on in the parahippocampal place area, so this is a very simple experiment that you can do. In fact, there's an undergrad in my lab who first looked at this problem. You can take the pattern of response. It looks very similar until you apply a stronger tool. You can apply some kind of supervised clustering. It doesn't really matter which clustering you use. You will always find that the number of canonical components in this part of the brain is 2. Okay, this is not from my lab. This is what people have even seen before. Okay, the challenge is to figure out what these 2 parts of the parahippocampal place area are doing, right? So let's do the most basic thing, right? You can look at, okay, let's take a large number of stimuli. We'll see what the top images are that these parts of the brain are responding to. Just by looking, there's a tradition in human cognitive neuroscience of doing this, so we did the same. Okay, here are top 5 responses of the first half of the PPA. Here are top 5 responses of the second half of the PPA. All of them are seeds. This is not a typo. The same scene appears 2 times. Now, you can look at this all you want. You can stare at this for a very long time. The point that I'm trying to make here is that these differences are really hard for you to discern. This is what's been going on for a while in this field, and nobody has a clue for what these differences are between these 2 parts of the brain, right? So we said, okay, let's take a different approach. And you probably already know where I'm going with this. You can take models. You can develop models for these 2 things, and then you can ask models, hey, tell us what the difference is between these 2 parts of the parahippocampal place area. So that's the idea, okay? Let's take it, because of time, I'm going to take some of these steps very quickly. This particular slide is just to show you that when you develop models of these 2 parts of the PPA, the models are very good, right? So you can predict the response of these parts of the brain pretty much to the noise ceiling of the data. Okay, so and then these models are also predicting something that's unique to each sub-region of the PPA. So that's the general idea here. So now you can turn that into a scientific question. Okay, if the models have really been able to capture the variance of responses in each of these individual bits, can you find images to modulate responses within the human PPA? So there are several strategies. I described a GAN-based strategy. Let me show you another strategy. So as I told you before, all of these models are image computable, right? So you can take any stimulus and you can run them through the model and you can get a prediction. So that's kind of what we did. You can take any stimulus and you can run it through the model and you find images, but this time you do something a little bit smarter. What you want to do is you want to engineer the kinds of stimuli that you find and you want to bin them into 2 broad groups. And the way you do this experiment is the follows. So you want to find 2 bags of images. I'm going to call these bags of images set 1 bag and a set 2 bag. But they are predicted to modulate the responses in the 2 parts of the PPA using something that's called a double dissociation, which means that for one subregion, you have a higher response for set 1. And for the other subregion, you have a higher response for set 2. Right? So everybody get this general idea? So you take 2 bags of images, you want to make sure that you are able to drive the 2 parts of the PPA differentially. So that's the general idea. Okay, so the way to think about this is that now we are using models for a different kind of purpose. So we want to set the response in the brain that we would like to see, right? And then we are going to use stimuli and models as the intermediary and identify the set of stimuli that will drive the responses of this part of the brain in a differential manner. So that's the big high-level description of what's going on here. Now, these are, of course, still model predictions, right? So let me first show you what these images look like. So once you do this entire test, what ends up happening is that you have a very different prediction for what these 2 parts of the PPA are going to do. right? So you can see this the second part of the PPA, which now seems to have very similar kinds of response features. And you can look at the PPA on the top, like these images on the top, and then you see a very different, just statistical properties of features in these two sets of images. We can worry about what these are, and we'll do that in the next few slides. But for now, the way to think about this is that would these two regions really modulate the two parts of the PPA differently? So we pre-registered this entire experiment. The idea is that we did things right. And here's how we're going to look at the data. Right? What we're going to do first is we'll identify the scene regions in every single subject. That's easy. You can turn on the brain. These parts in the black are what's the human PPA. The next thing that we're going to do is we're going to plot the difference in response for the two sets of images. Right? So why are we plotting the difference in response? We're lucky here that we have two groups, right? So if you plot a difference in response, you're going to get a positive number for this difference and a negative number for this difference. Right? Everybody see that? So this is just a visualization kind of aid, right? So if you just then plot that on the surface on the brain, you'll see something that goes from a negative response to a positive response. This is what you should see, okay? And when you look at this for groups of subjects, and you see a very canonical pattern, like you see almost every subject that you test. And these are not subjects on which the models were trained on, right? We wanted to make a claim that goes beyond this, like this is a canonical computation in each one of us. Each one of us has actually 2 bits within our parahippocampal place area. And now we can actually go inside and test those ideas more systematically. Okay. Now, so far what I've shown you is that, we can modulate responses, but what this still is very unsatisfactory, right? Because all we've shown you is that we can change the responses. We cannot tell you again about what these subregions are encoding. So what is going on in these two parts of the human PPA, right? Turns out that there's a lot of evidence for how to process scenes that comes from a number of, this particular idea of, navigation is important in other aspects of neuroscience as well as in robotics as well as in computer science, right? So there are a number of hypotheses that you can draw from. Okay, so which one do you choose? Here's everything that has been investigated in terms of hypotheses in the human parahippocampal place area, in both robotics as well as computer science, right? The good thing that we can do now is we can use models for hypothesis testing. In the first part of this talk, I showed you that these models are already so good that you can test all of these ideas on models directly without even having to run an experiment, right? So that's kind of what we did. And the one that really stands apart for now is this idea of scene content versus scene layout. If you talk to people in robotics who design robotic systems, they'll tell you that the two major things that a robot has to do is to first figure out, what things are in this space, and then wants to figure out, how it might navigate in that space. So this is like a canonical computation that people have wondered about for a while. So the way to test this experiment is the follows, right? What you can do is you can take a complete scene, and then you can break all the scene elements while keeping the features of the scene intact. So that's the general idea. You take a scene and you fracture the scene. And if you want to test for the content in the scene, the thing to do is you can add clutter. You can take a scene and you can parametrically add the amount of clutter in that particular scene. And when you do that and run that experiment on models of both halves of the PPA, you get a very different kind of pattern. What you find is one of the parahippocampal place area regions is very sensitive to whether a scene is arranged or fractured. Whereas the other half doesn't care about the pattern there, but cares more about the amount of clutter in the scene, right? And this gives us like a first estimate for what the two halves of the parahippocampal place area might be doing. So in general, this is a third kind of superpower that you have now because you have models, which is it can help figure out the organization of the human brain at a detail that was not accessible to us before, right? And based on that, in the first project, what we showed is that the dimensionality of the parahippocampal place area is at least 2. I say that at least 2 because we are still limited by the resolution of the data that we are working with. This is still fMRI. But then what we can also show is that these PPA subdivisions encode distinct information about scene content versus the scene layout. Okay, so at a very high level, I want to take a sort of big picture at this point. This is I don't know how many of you know about Hans Lukas Steuber. So Hans Lukas Steuber was a early psychologist turned neuroscientist. He was also he's the one who founded the Brain and Cognitive Science department at MIT. And when he talks about science in general, if you talk about the origin of how the scientific method works, there is there's a way of doing science which has a distinct set of trajectories. You can think of it from fire in physics all the way up to cognitive neuroscience, which is that you always start with explanation and then you move to prediction and then you move to control, right? For a long time, these three distinct phases of the scientific method were not accessible in psychology and neuroscience. But I think we are at a very exciting time today, which is that what I've already shown you is that if you have predictive models, you can go back and extract a bunch of explanations for the different kinds of things that exist in the visual system. But then you can also take these predictive models to get some handle at neural control. What I showed you was just the first attempt in this realm in human neuroscience, and there's a lot more to come. Okay, now, as I conclude, I also don't want to leave you with this general feeling that we've solved everything, right? There are a number of limitations using this method that we are already working on. One limitation, for example, is you may have heard of adversarial attacks. All of these models are also vulnerable to adversarial attacks. This is work that two graduate students in the lab who are both here have been working on. This idea that you can take a model and actually you can engineer a small kind of perturbation, and you can really drive the response of this particular image really down, right? These are at such a small scale that this cannot even be rendered on a good computer screen, right? So this should not change the responses, but we've actually already figuring out ways to improve models so that they don't have these issues. The second thing that we have been working on is, I started with just showing my appreciation for the organization of the human visual system. And the thing to note is that all current AI systems do not have any kind of organization, right? So what we've been doing in the lab now is we're trying to develop what we call as topographic models, and for vision, language, and the auditory system. And this is a paper that's under review. The punchline of this kind of work is that the neurons in the brain are not just strewn about haphazardly, right? Like there's a systematic organization in the brain, whereas when you look at the current AI systems, they look completely disorganized. So this is work that's being led by Mayuk, Menak, and Heather in the lab, and a bunch of our other collaborators. The goal is to try and see if we can take an unorganized system like the current AI systems and induce topographic arrangement while training so that the models learn something new and different as a way of making these models more high performing and efficient than they currently are. Okay, and so if you think of the space of neuro AI, so this kind of work is like taking principles that we know from neuroscience and plugging it back into AI systems. So that's the general idea for that kind of work in the lab. And as I conclude, of course, I would like to thank all the members of my very, very new lab, some of whom are here. My collaborators at MIT, and especially I would say like Freddie, with whom I am working on the PPA project, who's at the University of Edinburgh right now, and all our funding agencies. Thank you very much.
So much for the great talk. So I've been wondering since the first two sections of the talk that should I be surprised? So for example, the statistics of natural images is well defined. There is big literature, quantifying how the natural images look like. And then those models are trained on natural images. And then you are feeding them and testing them with stimuli that are based on natural images. So I should not be that surprised. And you answered part of this question in the last slide when you were showing that, oh, if I added a little bit of noise, then I can push the model way stray from what the original one is being. So could this just be all of it just an artifact of, oh, we trained a model on something that the brain was somehow trained on?
Yeah.
And
okay, so the question really is, should you be surprised? Is that the...
Yeah, more or less.
So it turns out that not all systems trained on naturalistic images will predict the brain equally well. So there is a version in which like you should be surprised. Because like if you take early like versions of these kinds of models, I mean things like AlexNet, for example. So that's a thing that I didn't get into very much in this talk because I wanted to keep like, significantly high level. Turns out that if you test those kinds of models on prediction, or especially the kinds of tests that I showed you in the second part of my talk, where all the old neuroscience studies, they do not do very well. They will fail. So it's not only about training on naturalistic stimuli. Now, the second thing you might do is say, okay, what else do you need training on, right? So think of these models as learning some kind of statistics. So the two things are interacting, right? Like you also need some kind of brain data to regress on, right? So it's a combination of both of these things. But when you test them even on things that the models had never encountered, things like line drawing stimuli, things like other kinds of noise. By the way, when I showed you that targeted noise throws these models off, you can take the same statistics of the targeted noise, but you can shuffle them around and the models don't care anymore. Okay, so there's something about the specific kind of noise. And that specific kind of noise can also tell us something about the brain. The kinds of features that the brains might be tuned for. That's kind of where we are leading with that kind of work. So it's not just about taking statistics from the natural world.
So if I trained on a model that is, like a variational version of the models that you started with, such that it's more, like robust against noise, would the plot that you just showed right there would have been better?
No, it turns out that so we've done that exact thing, by the way. Like you can take a model that's trained on noise, so which is called the robustified model, right? Turns out that those models don't predict brain data very well. So there is a trade-off. It's not that if you just have models that are more robust, your predictive accuracy keeps on going upward. So then those models are not useful anymore, right? Like if as soon as your models stop being predictive, then you don't need to work with them anymore. So that's another reason why you should not think that it's just about training on naturalistic stimuli. Cool. Question?
It's kind of like a broad question. We know that like parts of these, like the FFA can get, like certain people have lesions in the FFA and they have difficulties.
Yep.
Do you have any like thoughts on applications that you can use this for?
Yeah, that's exactly what we're doing right now. Is we have clinical partners who are who record from people with lesions in brains. We have other groups of participants who have other kinds of debilitating things like prosopagnosia. If you know prosopagnosia, it's the inability to discriminate between faces. So there are people in the world who cannot tell two faces apart by just looking at the properties of the face. So it's called prosopagnosia. Now, until this point in time, nobody knows why these atypical processing happens. Like we don't have a good idea. If you look at the NIH website, it does a very good job of being extremely vague. It's a problem in the occipitotemporal cortex. I'm like, yeah, where else in the brain will that happen, right? Like so, the reason is that each of these people has an FFA, right? So we don't know what's going on, except now that you have these highly trained and specific predictive computational models, you can tell the difference between a prosopagnosic brain and a regularly developing brain. So it's too premature for me to talk about right now, but yeah, I'll tell you more soon. But yeah, that's exactly where we're headed, like figuring out, what happens in people in lesions, and yeah, that's why I got into this in the first place.
That's like almost getting to control, because like you can modify the network and then try to get it. Yeah.
So you show that picture where they are the prediction of the faces with the fusion model. Can you please bring it back? I just want to make just curious for some of those faces. They look like from Picasso or Goya, very disturbing.
Oh, okay. I know what you mean. Yeah.
The landscape that you show below, pretty sure they were also, maybe they also are distorted, but they were not. To me, they did not bother me. Yes.
Yeah.
See,
These bother you more than this one?
Yes.
Yeah.
So I wonder. It's just me that how is missing harmony there? Is right. What is missing that the diffusion model
Yeah, that's a great question. So the thing I want to say is
I mean, the landscapes, I mean,
He-
I'm pretty sure they also have similar distorted features,
They do.
but they don't bother me. You know, I mean, but some of those things up there, I mean,
So there are a lot of these perceptual effects, by the way, in the literature. So for example, one is, I don't know if you've seen these faces with multiple eyes. People don't like those images at all. They don't like, I mean, there's an entire Reddit thread on this, which is, don't ask me how I got there, but you can go on that Reddit thread and you'll see like all these things with like 2 eyes, 2 noses and so on. Now, but let me be more specific about these sets of images, right? These are coming from a GAN with a generative adversarial network. Now, turns out these GANs are not as expressive as some of the new generative models.
so when you say expression, it means harmony.
No, in terms of, like how canonical the images are going to look like, right? So we want to decouple two things, which is the performance of the model versus the performance of the generator, right? So in this case, the generator is an old version. But if you look at some of the new versions of this that, I mean, I'm sure my students can show you very quickly, they are better looking if that's what we're going for. Now, we've run experiments with these kinds of stimuli in the brain. They do actually elicit a very high response in these parts of the brain. So there is evidence that even these images that look weird to you do drive responses in the human brain. Yeah.
One more we have online.
Yeah.
From your colleague Blue Me. She said, "Thank you for the insightful talk. I'm curious about how many data is needed to make the model could really generalize, such as to replicate the success in the foundation model, like the large internet."
Yeah. So actually, if you think about this, we are taking exactly the opposite approach, which is like the idea of the foundation model is to train the data on as much data as possible. We're trying to see how well we can predict as many of the data as possible using the minimal amount of brain data as supervision, right? So we've not done those exact tests, but our intuition right now is that the quality of data matters more than the quantity of data. And turns out that you need very few samples to predict a large number of these kinds of things that you're seeing, even if you want to develop like completely in silico models. So now there are other aspects of this which we can take offline, because you can ask, okay, how many data do we actually really need to train the neural network model? And that's another question. We've not explored those as well right now. But yeah, it's completely within the space of things that we should look into.
Give like an order of magnitude, like millions, billions. How many data points?
For the neural network training or the brain or this model training?
It's model training.
Thousands, less than thousands.
So when it's convolutional neural network, now on the first layer is the layer, they basically just see the small features. And then they go from small to large scale recognition. That's how the brain, that's how we do the human mind.
Pretty much. So the thing is, at some level of abstraction, I mean, there are things that the human brain does not have that models have. Some algorithms like weight sharing, I mean, we can talk about.
But artists, they do the opposite.
Yeah.
They go from large to small scale.
Yeah.
In order to represent a landscape, they first try to capture the big main patches of colors, and then slowly, they.
Yeah,
No.
we are trying to unconfound two things. Whether we're trying to unconfound inference from like synthesis, right? When you do synthesis, then you do want to care about the gestalt first. Like when you draw, right? Like you can think of it as you care about the large picture, the big picture before you get into details, right? But when you're talking about inference, which is like all the computational stages, artists are pretty much similar to us. People have also like studied them in quite a bit of detail.
But these are natural what artists do at this point.
Yeah, but that's what you do as well. But so that artist thing that you just talked about is the synthesis procedure, not the computational procedure. That's the difference.
This is past now, so let's stop here.
Okay.